My name is John Draper, and I'm the Program Director for Research and Partnerships at the Stem Cell Network. And you're joining us today for the Cryopreservation for Regenerative Medicine workshop. Thank you very much for registering and taking part in this event. The workshop booklet is available, and I'll just drop this right now into the chat, or I'll do that in a moment. Here we go. This is a webinar, so there is a Q&A function that you'll be able to use to ask questions, and there will be dedicated Q&A sessions immediately after each of the talks, but also at the end of the talk sessions as well. So please do address your questions to the speakers today. It will be really, I think, useful, start conversation, and they will address those questions as they come in. The breaks are scheduled, so there's a break here about 3:25 p.m. Eastern time. There will be a break which will last about 20 minutes before we move on to the second session as well. This workshop is going to be recorded as well, and we will make the recordings available at a later point. So if you miss something, there is an opportunity to catch up with it later. But your participation is valued, so please do take the time to interact with this opportunity to discuss very important techniques and principles of cryopreservation in regenerative medicine. At that point, I'm going to hand it over to Kate Murray, who is the Executive Director and CEO of the Stem Cell Network, who will provide a few words of introduction and welcome. Thank you very much, Kate.
Good morning. Thanks, John. Afternoon or evening, depending on where you are in the world. As John mentioned, I head up the Stem Cell Network. And first, I'd like to convey the best wishes of Dr. Michael Rudnicki, Stem Cell Network's Scientific Director. He's unable to join today due to a scheduling issue, but he's thrilled to have so many of you gathering for this event. So we at SCN are really proud to be partnering with the Society for Low Temperature Biology on this initiative. We've got well over 170 people registered for this session and the June session. You come from all over the world, 28 countries in fact. Your areas of interest span the continuum of research, from basic research, preclinical and clinical research. And approximately a third of you have a specific interest in biomanufacturing, an area of focus that now, thanks to COVID, even my mother's interested in knowing about. So for those who are not familiar with the Stem Cell Network, let me give you a few highlights about us. SCN is currently celebrating its 20th anniversary. We are Canada's only national academic network for researchers and trainees who are using stem cells to fuel the field of regenerative medicine. We partner with industry, with charities, and of course the university sectors to support leading edge research. And when SCN first started, it was a small group of about 35 PIs from across Canada and their labs. Today, we have approximately 200 labs that make up the network, coast to coast. We've provided 118 million plus into research and training. We've supported 3,300 HQP with their learnings. We've supported 24 clinical trials and seen nearly 24 companies created or enhanced thanks to SCN research funding. I'm thrilled to be able to share with some of you that may not know that the Government of Canada has just reiterated its trust in SCN and made a commitment to stem cell and regenerative medicine research for the future with a new investment that more than doubles the annual budget of the Stem Cell Network. This is great news for Canada and for the contribution that I know our science community will make on the international stage. But it's not just me who feels positive about the sector, so do investors, and it's really an exciting time for regenerative medicine globally. The Alliance for Regenerative Medicine, based out of the United States, reported sector financing reached just under 20 billion in 2020. That's US dollars, of course. That is doubling from the previous year. And now they're predicting that 2021 will be another record year for the sector. But this momentum really only happens because of meetings like this, where the community gets to gather, share best practices, learn, and network. So I'd like to congratulate my colleague John Draper and our dear friend Dr. Glenn Stacy for your leadership in pulling together today's event. I know it's going to be a really impactful and valuable day for many of you. And now it's my honor to turn the floor over to Glenn, who's the past chair for the Society for Low Temperature Biology.
Okay, welcome everybody, and thanks for that introduction, Kate. We've been working with Canadian Stem Cell Network for some time on this now, and thanks to John and Kate for their enthusiasm and for the interest of the Canadian delegates as well. As Kate said, I'm the ex-chair for the Society for Low Temperature Biology, which is a registered charity supporting in promotion of communications amongst scientists and education in cryobiology. And I should introduce the person who is the current chair for the Society for Low Temperature Biology, Christiani Amorim. Over to you, Christiani.
Thank you, Glenn. Sorry for my delay. Good morning, good afternoon, or good evening, depends where you are. I'm very happy to welcome you all to the first day of this workshop. And it's a great pleasure for me to be part of this event that has been brilliantly put together by Dr. Draper and Professor Stacy. So as Glenn mentioned, I'm the current chair of the Society of Low Temperature Biology, or SLTB, and I would like to take this opportunity to say a few words about our society for the ones that are not acquainted with it. So the SLTB, it's a quite old society. It was funded back in 1964, and the main purpose it was to promote research into the effects of low temperatures to all type of organisms and their cells, tissue, and organs. So as we know, such studies have a huge variety of applications in numerous fields, from biology and medicine to engineering, conservation, and environmental science. So for the ones developing research on cryopreservation, I highly suggest you to become a member of our society. In our annual meetings, we learn a lot. We have this huge variety of topics related to low temperature biology, and we have a very enthusiastic and informal atmosphere. I'm very proud to say that our meetings are really more like meeting friends than a formal conference. So our next meeting will be in October. I would invite you to participate. Due to the pandemic, it will be online again. But last year, our virtual meeting, it was a great success. We had people all over the world. We have a huge interaction. It was very nice to see old colleagues as well. So if you want to know more about SLTB or to want to know more about our annual meetings, I would like to invite you to visit our website, www.sltb.info. I think that's it.
Okay, thank you very much, Christiani. So we will move straight on to the very first session, which you're also going to be chairing as well, Christiani. So with that, we will start with the fundamental principles of cryopreservation. Thank you.
So now I'd like to, in this first section, I would like to introduce the first speaker, Professor Devireddy, who will give a talk on the fundamental principles of cryopreservation of animal cells. Professor Devireddy is a DeSoto Parish Chapter University alumni professor and the Louisiana Land and Exploration Company endowed chair professor of mechanical engineering at Louisiana State University in the United States. Professor Devireddy's primary contributions include developing new methods to assess the state of water during freezing in biological systems, to fabricating novel micro-scale biothermal devices, and more recently, Dr. Devireddy also has been directing osteogenic differentiation of adult stem cells using microRNAs. He has received numerous research and teaching awards. He has been an advisor for several PhD students and postdocs, and he also serves on editorial boards of several journals and has also been editing book chapters. So I would like to invite you, Professor Devireddy, to start your talk now.
Thank you, Christiani. Can you, I guess, I can't, if it is an actual presentation or ask if you can hear me, but I'm assuming you can hear me. So the title of my talk is, like Christiani mentioned, Fundamentals of Cryopreservation. And hopefully by the end of this talk, you should have a sense about three different things. What happens to cells when we freeze them? How does this process be impacted by the type of the cell that you're freezing? And how do you measure these processes? How do we model them? And how can we use this information to better develop what I would call optimal cryopreservation or rationally designed cryopreservation protocols? Before I start with talking about cryopreservation, I want you to realize that this is just one of the three major different methods of techniques available to store tissues or cells. So what you see here is there's one which is freezing and cryopreservation, which is essentially what we're going to be talking about. There's also freeze drying. Essentially, you freeze up to you start off with a liquid, you freeze it to or ice forms, and then you remove all the water, right? And essentially, you get to a glassy state. Or you can also do simple drying. Essentially, you take a liquid and you dry it all the way to the glassy state. All of them have their advantages and disadvantages. But the focus of this talk is to take a liquid and to get it down to a low enough temperature that we can store it for long periods of time without the cells getting damaged, which is essentially cryopreservation. Now, why do we want to study cryopreservation? We want to study cryopreservation because animals do it. So we always want to learn from nature. And there are lots and lots of animals in nature and species that essentially can survive freezing and come back essentially once the weather warms up, so to speak. In Canada, we have this wood frog, which is a Rana sylvatica, which is absolutely amazing. In winter, it essentially becomes a solid block of ice. I mean, I've handled these things. They essentially crack. I mean, so they're essentially fully solid frozen. But come spring, they're jumping around. And all of these species, right? I mean, so some of them can be alive for as long as 40,000 years. So this is not just a temporary event. So we can store these things for long periods of time and we can recover them back. So we want to basically, since animals can do it, maybe we can mimic what is happening in nature and do it in the lab, right? Oops. So how do animals do this in nature? Almost all of them do this by creating some chemicals within the body. For example, the wood frog creates glucose and sucrose. And they're also very smart. They don't nucleate ice or they don't form ice during freezing next to major organs. They form it what I would call, innocuous places. But on the next talk, you'll hear Dr. Elliott talk about what happens to these cells when you expose them to all these cryoprotective agents, how to model it, and how do we optimize the process. And I just wanted to know that these cryoprotective agents there are more than, at least as far as I know, 60 to 100 different chemicals that have been used in the literature, and they've been shown to various degrees to improve the cell outcome, cell preservation outcome. Okay. So I always get this question when I tell engineers I'm doing, cryopreservation. The question I always get is, can we freeze Austin Powers and thaw him back up again? Like they show in the movie. I guess the simple answer is yes, we can do it for most cells, to some tissues to a certain degree, but I would say not to full organs yet, although that is changing quite rapidly as we speak. So why can't we do it? What happens to cells when we freeze them? So lots of things happen. First thing that has to happen is we have to add the chemicals, like I said, right? And when we add chemicals, well, I'll show you briefly what happens to cells when we add chemicals, right? And that, we call that as osmotic injury, right? Basically adding chemicals causes some damage. And then you have to cool the cells from whatever, 37 degrees or room temperature to 4 degrees. And this also causes what is known as chilling injury, right? And this is very prevalent in embryos and oocytes, which are very, very sensitive to chilling injury. And then the major part of this talk is going to be on freezing injury. What happens when we cool these cells from 4 degrees Celsius to about -40? Once you get to -40, almost all the freezing processes are done. There's very minimal damage after that. And then you have to store them. You got to store them below the glass transition temperature. And if you do it right, the damage mechanism is minimal. And then after you store it, you want to use it, which means you got to thaw it back up again. You got to heat it from wherever you're storing it to the room temperature. And that again causes injury because depending on how you thaw it, the ice nucleation can be damaged. And then you have to remove the chemicals. Most of these chemicals that we add are not something that the body wants to use or body feels happy having inside oneself, so to speak. So we have to remove them. And removing these CPAs causes essentially the same injury that we started out with. And then, of course, you have to transplant it, check for the immunological response. So there's lots of little things that have to go right, and a few major things that have to go right for a whole process of biofreezing or cryopreservation to be successful. So what happens to the cells when you add cryoprotective agents? It's a very, very simple schematic, and I'm just going to focus only on the basics. So assume a cell, right, essentially sitting salt water inside, salt water outside, and then you add all these red dots, which I'm essentially calling as a chemical. When you add chemicals outside, you're essentially messing with the potential. So the water concentration outside is less than the water concentration inside, which means now the water wants to come out. This is essentially osmosis, right? And we see it, this is one way you desalinate, for example, right? And once you have this water coming out, the cell tends to shrink. So you see the cell volume with time, it shrinks because water is leaving. And after a period of time, all these CPAs, now assuming it's a permeant CPA, there are others that don't permeate, but assuming this CPA can get into the cell, these red dots or orange dots get into the cell and the cell volume goes up again. Now, when you shrink a cell, the volume is shrinking, the membranes are coming together. And if you shrink them too much, right, they fuse and then you can't bring them back up again. That causes damage, right? That's the shrinking response. And then when you're swelling it, right, if you swell it too much, same thing happens. The cells don't want to expand before this, they blow up, so to speak. And that causes, swelling. So this process is a shrink swell curve. And, and it also, the longer you do this, the more time you expose the cells, the more damage you're doing, because most of these chemicals are not things the cell is used to, living with, so to speak. So that causes damage. So your problem now is to minimize the cell volume changes, minimize the time, and how do we do this efficiently? There are models to do this, and I briefly got a chance to look at Dr. Elliott's slides before this thing started, so I know she's going to talk more about this, and you should have a better sense of this once we're done with that talk. But getting ahead to my talk, which is freezing, what happens to cells when you freeze them? So again, we have a cell, right? And then when you freeze cells in a solution, imagine this room is full of cells, right? And you start freezing this room. More often than not, 99.9%, the nucleation of the ice, which is a statistical event, happens not inside the cell, but outside the cell. This is purely statistical. It doesn't matter what population of cells you have, unless the cell concentration density is insanely high. We're talking about, which is actually not, which rarely ever happens. About 10% of the volume is taken by cells, 90% is non-cell volume. Given that, the probability of ice, you can't quite control where it happens, it's liable to happen outside the cell. Once that happens, right, once ice nucleates, now I'm not sure how many of you know this, but when you freeze salt water, the frozen fraction is always pure water. The salts get rejected. So if you ever lost in an island, for example, right, and if you want to get fresh water, and if you have access to unlimited power, the thing to do is take salt water and freeze it, right, and eat only from the edges, so to speak. Leave the center in the middle where all the salts get rejected. So something similar happens here. So when you form ice, like I said, is pure water, and you have this, now that water has formed ice, the remaining salts have less water volume to occupy, which means their concentration goes up. Once their concentration goes up, now, and then you start cooling them, right? Three things can happen. One is a slow cooling, so I'm cooling them slowly now. Once this happens, the water inside the cell wants to come out because, like I said, water concentration outside is more, so potential is less. So water wants to go from low concentration or high potential to high concentration or low potential, which means the cells tend to shrink, right? We call that as a cell that I call that as a cell that becomes a prune. So imagine yourself sitting in a vat of salt water for 24 hours, right? And then it happens to us when you take your bath for too long, your fingers all wrinkle up. Something similar happens when you do this slow cooling. On the opposite side, if you cool them very rapidly, right, the water doesn't have a chance to come out of the cell and it forms ice inside. We call that as ice formation inside the cells, or fancy word, intracell ice formation, right? So essentially this is ice forming inside the cell, and this is water leaving the cell to form ice outside. Now, both of these things are bad. Now, like I said, self-dehydration or water transport is what I call a prune cell. When you have ice forming inside the cell, I decided to call that as poked. Basically, ice can be damaging. Ice occupies more volume than water, which means when it forms ice, it wants more space to occupy, and that causes damage. Somewhere in there, somewhere in the middle is the optimal cooling rate, right? And that's what you want to find. And this inverted U curve has been studied for a long time. It's well established, but you have two damage mechanisms. One, because you're exposed to salt concentrations long periods of time, and the cell is shrinking. Or the opposite, the cell is not losing water fast enough and ice forms inside, and that is damaging, like I said, because ice can be destructive if improperly formed, right? So same things happen. The similar things that happen in what you would call cells also happen in tissues, right? Here you see ice forming inside the cells. Here you see ice forming outside the cells, and this is an unfrozen tissue section. Now, why is this important, right? This inverted cooling rate looks very straightforward. All you got to do is find the middle rate somewhere, right? You got slow cooling, you got fast cooling, find the middle one, and we're set. But the problem is this cooling rate, which I've shown on this axis. Now, notice that this axis is on a log scale, which means I'm going from 1 to 10 to 100 to 1,000 to 10,000. So red blood cells, for example, are sitting all the way down here, whereas bone marrow cells are sitting all the way down here. So your cooling rate, optimal cooling rate, so to speak, goes from all the way from as low as 1 degree to 10,000 degrees C per minute. So we have a huge range of cooling rates to decide to find the optimal cooling rate. And again, I also want to emphasize that this cooling rate, this optimal cooling rate, can be modified if you change the solution the cells are freezing in. So not only is it a cell dependent, it is also dependent on the media within which you're freezing these cells. So how do you measure these processes, right? The simplest thing to do is to observe it, right? Back in the 1850s, Molish and his colleagues, Muller, so they essentially they took a microscope, they took a whole microscope, and they put it in a box of ice. Now, if you add salt to ice, we can lower the temperature, right? So essentially, by adding more and more salt to this ice, they're able to put these cells under different temperatures. You can then take images of it, right? And it's amazing that those measurements back in the 1850s are essentially what we still use to define the biophysical process during freezing in cells and tissues even now. But we don't quite use, I don't quite take a whole microscope and put in ice. We do a bit more expensive. What we have is a cryo microscope. And essentially, imagine you put a cells here, it's basically, think of it like a heated stage. So what you have is a stage, and then you have liquid nitrogen that you pass through it, right, under the stage. By controlling the amount of heating, you can control the temperature experienced by the cells. So you have a heating, and then you can supply liquid nitrogen, and you don't control the liquid nitrogen. We control the heating because that's easier to control. by deciding how much energy I'm supplying in or removing. You can put cells in here, right? You can watch them on a microscope connected to a computer. We can take videos. We can impose different cooling rates because by depending on how I'm heating, you can change the cooling rate. And this allows us to make movies and visualize the process. For example, here is an example of mouse or oyster embryos being cooled at 10 degrees C per minute, right? You see that initial first wave, oops. The first wave, you watch now again, the first blackening is essentially ice formation, which is outside. And then after that, you see the cells blackening. And the cells blackening is ice forming inside the cell, right? Ice is opaque to light, so it looks like us to their black things. And we can model this, you can predict, you can plot what fraction of the cells experienced IAF at different temperatures, right? And essentially now, knowing what we know, right? Just from this one simple experiment, we can predict immediately, right? If I want to optimally cryopreserve these cells, I should be cooling them at less than 10 degrees C per minute. So essentially, we have an idea now, the cooling rate I can go from is 0.1 to about 10 degrees C per minute, which is a lot better than what I started out with, which is 1 degree to 10,000 degrees, right? So we can also, we can also, like I said, observe these cells under the microscope. Here is an example of a cell that is shrinking in volume. Now, the cells as a shrink reach a final volume, which is called as V sub P, which essentially this represents all the cell volume that is not water, right? That's the lowest the cell can shrink because, there's only, the cell is not just water, it has other things in it. We can look at these images. We can plot them again as a function of the original volume to the volume at different temperatures. So I'm plotting this. V is the volume at a given temperature. V naught represents the initial volume. We can plot that on a y-axis as a function of temperature. And again, we got to be careful here though, right? I'm only getting two-dimensional images. So we have to use some kind of a correlation between cell volume and surface area. Essentially, we are assuming the cells to be spherical. That will allow me to convert what are essentially 2D measurements into volumetric measurements, right? And we'll get to these numbers in a second. So this allows me to figure out how fast a cell is shrinking or changing its volume for a given cooling rate. And again, remember, the cell is shrinking because there are differences in salt concentrations between outside and inside. So we can make these measurements. There are lots and lots of these measurements for lots and lots of cells, right? And we can do it without a CPA. We can do it with different CPAs, for example, glycerol, Dmso. These are all the same type of stem cells, and they have 3 different CPAs. And if you see the behavior of the cell, right, how the cell changes its volume at the cooling rate is different, which essentially tells us that by changing the media, I can affect how the water will leave the cell, which means I can now affect what the optimal cooling rate is for the given cell, simplistically speaking. So similar things happen, like I said, in tissues, but measuring these in tissues is a little bit more complicated because we cannot visualize them because these are tissues essentially are opaque to light, so microscopy doesn't quite work. So we do something different. This was built back in the 90s on Boris Lubinsky's lab, I think. It's a well-established technique. So you have a high temperature and a low temperature. You see these 2 copper blocks are a high temperature and low temperature, and you maintain them at different temperatures. And you have a glass slide that you can push across these 2 blocks, depending on what the gap is and how fast you are pushing them. You can impose at different cooling rates, right? So depending on the gap and how fast you're pulling it and the temperature difference, you get a cooling rate that you can impose on whatever the sample is that is sitting on the slide. For this tissue section, once you get to a temperature, we slam freeze it. Essentially, we take a snapshot of the tissue at that temperature, and then we can section it. We can then image it. Here's an example of rat liver tissue at different temperatures, 0 degrees, -4, -8, and 20 degrees. All of this was done at 5 degrees C per minute. And as you see, as I go to lower and lower temperatures, you see more and more white spaces. Essentially, these white spaces are eyes, so which tells me you can work backwards now. So you know, as the vice volume is increasing, you know how much the cell volume has shrunk. We can plot a similar graph for tissues that we were able to do for cells. Although this is a much more laborious process. This takes 3 to 4 weeks, as opposed to microscopy. We should be able to get that result in a day or two. So how is this data useful? As engineers, one of the things we want to do is make measurements and use models, use those measurements to get some model parameters, and then use those models to predict things that we can't measure, right? That's the things that you want to be doing as scientists. You want to make some measurements, and then you want to use it to make predictions that are not quite plausible with current techniques, so to speak. So simplistically, right, here is again my cell, and I have this water that is leaving the cell, and I have this ice that is formed outside. And this change in the cell volume, the volume dV, the change in cell volume with respect to temperature, which is dT, that's the change in cell volume, is equal to the permeability of the water to the cell, obviously, right? If the water can leave the cell faster through a porous membrane, right, it'll leave faster. If the membrane is not as porous or not as quite happy to let water through, it'll have a different value. So the cell shrinking will depend on this LP, which is permeability of the cell, right? Of course, the area of the cell, the more area you have, the more water can leave, right? And obviously cooling rate, the faster you're cooling it, the less time it has, which means the less time for the volume to shrink. And what is driving this? What is driving it is the potential difference from the potential of the water inside to potential to outside. And this potential is inversely correlated to concentration. And we can model this somewhat slightly differently because this permeability, what we found from experiments is that LP itself changes with temperature. So this permeability of the cell doesn't remain the same at say -1 degrees to -2 degrees to -3 degrees. So to account for that, we use the Arrhenius model. So this LPG is a reference value at 0 degrees. And then we have this ELP, which is an activation energy, which tells me essentially how fast is this permeability dropping as I go down temperature. So now LP itself becomes a function of 2 parameters, a reference permeability at some temperature, essentially TR, and the ELP is the activation energy, which tells me how fast this LP is dropping as I cool down this, as I drop the temperature. So once I make these measurements, I need to figure out what these 2 parameters are. So if I make a measurement, if I know what dV over dt is, that allow me to get LPG values and ELP by doing some curve fitting. There are a bunch of assumptions in this model. Some are large, some are medium, some are really small. But in terms of engineering usefulness, these models give you numbers within 10% or maybe 20%, which allows us to be essentially bracket the experimental matrix that you have to do, right? So here is an example of, so this LPG and ELP, the thing that I want you to pay attention is that these are not just independent numbers, because I'm only curve fitting data to one set of data. So there's a combination of this LPG and ELP that will give me one LP value that will then fit to this data. Here is an example. So I've plotted LPG or ELP for same cell type, but that's a stromal vascular. Forget about the red dot. So these are 4 different cell types, right? I'm plotting LPG as ELP. And here is an example of, with every parameter, every combination of ELP and LPG within this curve, right? Within this curve will fit the data for this cell with no CPA with about 98% degree of confidence. Same thing for glycerol, same thing for Dmso. Again, notice the LPG and ELP have changed when I add CPA, when I don't have CPA. And this is also not just a function of, CPA, it's also a function of cell type. If I go to a different cell type, if I go to a different cell type, and if I go to a different cell type, you see that the curve, the space within which the combination of these parameters is changing. In fact, these are essentially not, although as I said, they're different cell types, they're essentially all stem cells, except they're different passages, right? Think of this as stromal vascular fraction, which you get from the initial fraction. That's passage 1 or passage 0 after two days or 4 days. Passage 2 is, we have passage 1 and we have passage 2, and this is passage 4. These are all stem cells, and yet you will see that as they differentiate or as they double up, you will see the permeabilities are changing. So it is not just different cell types, even if you have the same cell type, for example, stem cells, and if you culture them, the culturing time point also will affect how best you have to cool them and freeze them if you want to store them, right? So the things to learn so far, right? The cell properties are dependent on the cell type, right? They're also dependent on the media they're being frozen in, right? So two things to worry about, not just the cell type. So here is an example of, we can now take those numbers that we have, all the ELPs and LPGs, and we can predict optimal cooling rates. And I'll tell you in a second how we calculated this. But the attention that I want you to draw is same cell type, different media gives me different predicted cooling rates, right? Again, you passage the cell for 4 days, you get a different cooling rate. Do it twice, 2 more passages, and 2 more passages. So not only is it the cooling rate, but you also have to worry about the CPA and the cell type. Now here I'm just plotting the same data, except I plotted them in terms of the passages to show you if you have no CPA, passage 0 requires a completely different cooling rate than 10% glycerol or 10% Dmso. So how do we predict these cooling rates? The traditional way used to be that, let me back up a little. So here you have this. Now, as you start cooling this cell at different cooling rates, right? At a faster cooling rate, I'll follow a curve that looks like this, right? At a faster cooling rate, I'll follow a curve that looks like this. If I cool it really, really, really rapidly, the volume will not change at all. Essentially, you get a horizontal line. So what is happening when this curve gets cooled down? So up to this point, let's say I have a curve that goes like this. The difference, that change in that graph essentially is water that is sitting inside this cell. And if you cool it further down, that water has nowhere to go but to form ice. So we decided, it's almost arbitrary definition. We decided I only want to have 10% of the water volume to still be in the cell volume, right? When all the freezing stops. Essentially, I'm saying when I get to this temperature, of whatever, minus 10 degrees, I want 10% of the water to still be in there, right? And we define that as optimal. Basically, we're trying to find a cooling rate that's a little bit farther than just slow cooling, but not so close to ice formation. So we took all these parameters, and then we decided to model them with the model that we used. We plotted all of them as a function of activation energy, and then optimal cooling rate that we decided to call as, doesn't matter, up G over CRC. And then we plotted this curve here, and then for a bunch of cells that we could find. And you will see that almost all cells, human sperm to hepatocytes to lymphocytes, they all follow reasonably well with this curve, except tumor cells. And tumor cells are a different beast altogether. And again, we don't want to cool, we don't want to, unless you're doing research on tumor cells in a lab, if you're sitting them in a human body, I want to kill the tumor cells. I do not want to cryopreserve them. So this is not such a bad thing. So you can think of this as optimal destruction rate for a tumor cell. Essentially, if once you get the LPG and ELP values from the model, we only have to worry about one other cell parameter, which is the surface area to the water volume. Remember, like I said, the larger the surface area of the cell, the more ability for the water to pass through, which means my B optimum can be higher. And if I have the more water volume, it requires more time for it to leave, so inversely proportional to my optimal cooling rate. So once you do those experiments, those measured experiments using either cryo microscopy or using the certification stage, we can model this ELP and LPG. Once we get those numbers, plug them into this equation that will allow me to get my optimal cooling rate, right? So what have you learned? Did I go over? My watch is saying 4:40. So looks like I went over a little bit. So what have you learned so far? Hopefully you learned that when you freeze cells, 2 things happen, right? You got to worry about slow cooling injury. So you become a prune, or you become a fast cooling injury because you become poked, right? And how we model these things, I talked mostly about osmosis, water transport, but there are similar models for intercellular ice formation. They're a little bit more detailed, more involved, but essentially physics remains the same. You're trying to model a physical, we're trying to model an observational measurement of how fast ice forms inside the cell. And it also has 2 different parameters. And you can essentially work backwards from ice formation to optimal cooling rate, like we did from water transport to the cooling rate. And hopefully, like I said, you figured out that optimal cooling rates are not just one number. There's a range of numbers, and that depends on the cells. It is morphological, and it's also physiological in the sense it depends on how you're freezing these cells. And finally, right, we can't unfreeze Austin Powers yet, but hopefully at some point we'll be able to do it. So, you have any questions?
Thank you, Ram. It was a very exciting talk, as usual.
Thank you.
We'll leave all the questions for the end of the session. So
Okay.
please feel free to write all your questions in the Q&R, and we'll read them and discuss them later on at the end of Dr. Hunt's talk. So now I'd like to introduce our next speaker, Dr. Gloria Elliott. Dr. Elliott is a professor of mechanical engineering and engineering science at the University of North Carolina at Charlotte, and director of the Biostability Lab. Her research focuses on material and process innovations to achieve advances in cell and tissue banking. And Dr. Elliott will talk today about cryoprotectants, balancing toxicity and efficacy. Dr. Elliott, the floor is yours.
Thank you. Well, I won't go through that introduction again, so I'll just skip along to what our learning objectives are for this particular talk. So I was tasked with trying to teach you the different types of cryoprotectants and their mode of action, to appreciate the diverse biological effects of DMSO, the balance of toxicity and benefit, and how we can go about reducing DMSO toxicity. And finally, I was asked to talk to you about how we carry out cryoprotective toxicity testing in order to achieve good outcomes. So along those lines, I'm just going to hit on the basic methods, and then I'm going to kind of leap ahead and talk about where we could go. Okay, so as a way of sort of dovetailing from what Dr. Devireddy just presented, I just wanted to recap these injuries. Actually, I'm going to take a quick pause. I see the chat lighting up, so I want to make sure everyone's viewing slides okay. Okay, I see those are for the previous speaker, so I'm going to ignore that and keep going. Okay, so to recap what Dr. Devireddy just talked about, he presented in great detail about how we arrive at these inverted U viability curves, and he talked about optimal cooling and the different types of injury that manifest. So intracellular ice formation at high cooling rates and excessive dehydration at slow cooling rates. And the premise for all of this is the rate at which water leaves the cell. So I've tried to capture the essence of a lot of complicated thermodynamic equations and just drill down to the essence of what a biologist needs to understand. So the rate at which water is going to leave a cell, the dVW with respect to time, is going to be driven by two major things. You have your characteristics of the cell, the membrane permeability coefficient that Dr. Reddy was just talking about, and features like surface area and temperature. And you also have your driving force, the difference between the intracellular concentration or the intracellular osmolality and the extracellular osmolality. When we freeze cells in solution, when liquid water comes out of solution and becomes solid ice, what happens is the remaining solution becomes concentrated in the solutes. And this is what sets up the driving force for water transport. So we saw that, this LP is very much dependent on the cell type and the membrane composition. And you'll see a lot of differences from cell type to cell type. And also the presence of cryoprotectants can also alter this value a little bit. But what I'm going to focus on in my talk is more how cryoprotectants affect this driving force, because ultimately this is what initiates the water transport in the first place. So when we have ice forming in the extracellular space and causing this concentration increase, a couple of things can happen. You know, we can, if the water transport is slow, you know, if we have small LP values or small or minimal surface area, water gets trapped. And so, as we know from the second law of thermodynamics, nature abhors a gradient. So this is why Humpty Dumpty falls off the wall and your tires deflate when you run over a nail. It's why your coffee cools, and it's why cells undergo osmosis. They want to equilibrate that gradient in chemical potential or osmotic pressure, osmolality. Those are all roughly equivalent between inside and outside. So if water transport is slow, intracellular ice will form, concentrating the intracellular solutes and making everything balance between inside and outside. If the LP is high or lots of surface area relative to, to the cooling rate, the cell will dehydrate extensively. It'll move water quickly to concentrate that intracellular solution. So the thing we need to worry about up here is whether we exceed what we know to be these critical limits for the cell. There's a critical volume limit beyond which you run into irreversible changes in the cell, that you can have membranes folding in on each other, coalescing, membrane deletions, etc., such that when the cell rehydrates, we have a different cell. It's a leaky cell. So to recap, then this is how we end up with the inverted U, and we have high viability where each of these injury mechanisms is minimized. And this inverted U is going to look different from cell type to cell type. When we add CPAs into the mix, we're trying to ameliorate injury on both sides of the inverted U. And the outcome is that we get an increase in survival across the board, and we have a lot more flexibility in terms of the cooling rate that we can use to preserve cells. So I'm going to talk a lot about this driving force as we go forward. I think you now have an appreciation that CPAs can alter this somewhat, but this is also a big deal, this driving force as well. Okay, so let's get on to the learning objectives of the course, or of the lecture. Key types of cryoprotectants. I've batched them this way: permeating CPAs, non-permeating CPs, and what I call enhancers. This is the boutique. These are the boutique items down here. So permeating CPAs like dimethyl sulfoxide, glycerol, ethylene glycol, basically enter the cell. If you were to put cells in suspension containing a penetrating CPA of a certain concentration, it will move into the cell until that chemical potential gradient is equilibrated. By contrast, non-permeating CPAs don't enter the cell. They're too big, and there's no way for these molecules to get through the cell membrane. So if you think about, and there are many that fall into this category, sucrose, trehalose, mannitol, for example, in the sugars, hydroxyethyl starch, PVP, PEG, there are lots more in the polymer category as well. But bottom line is, if you start with a cell containing, roughly 300 milliosmol physiologic salts, and you drop it into a non-penetrating concentration of, let's say, for example, 1 molar, what's going to happen in this case is the cell is going to lose water, decrease in volume until the physiologic salts inside the cell are concentrated to a 1 molar level, and this gradient is equilibrated. So all of these things are thermodynamically driven processes that are obeying the second law of thermodynamics. Enhancers, what I've lumped into this category is everything that kind of isn't based on these features of osmotic regulation. So for example, antioxidants, apoptosis inhibitor, these are ways of biochemically intervening with a cell that's under stress or has begun a death cascade, and these are rescuers, if you will. There are other additives like antifreeze proteins that can modulate the temperature at which ice forms, or the shape and morphology of ice crystals, making them more benign, etc. There are things that you can add into the mix to enhance the TG, and these compounds fit into that category, or increase the viscosity, or use assert some physical effect, that biophysical effect that minimizes ice formation. And these are fluid boundaries, because if you take, for example, trehalose, which doesn't penetrate the cell, but you use some exquisite technology to allow it to penetrate, then it now falls into this category. And many of these non-permeating CPAs also have some what I would call boutique effects as well, in terms of modifying TG and enhancing viscosity, and providing specific protection to biomolecules. Okay, so with that in mind, let's get right over to digging into the details of how these things work. So permeating CPAs at a most basic level reduce the volume of freezable water, both inside and outside the cell. Dmso is an example of a superior cryoprotectant. Many of the better cryoprotectants hydrogen bond with water and produce hydrogen bond strengths that are stronger than the water-water bonds. And what that does is basically make it difficult for water molecules to see each other and to form that ice phase. So you can see in the first figure here that in the presence of increasing amounts of Dmso, the amount of ice that's formed in these solutions at progressively lower temperatures is less than in the absence of cryoprotectants. So less ice inside the cell means less probability of intracellular ice formation, and that reduces what we call the fast cooling injury, or the IIF injury. But the fact that we have also put things inside the cell, so we've now increased the concentration of the intracellular space. Because of that, when ice does form outside the cell, there's less of a gradient that has to be equilibrated. Less of a gradient means less water has to move from inside the cell to outside the cell in order to reestablish equilibrium. So that means there's less risk of cell dehydration and exceeding those critical volumes and inducing osmotic damage. The fact that less water has to leave the cell also means that of the solutes that are inside the cell, these physiologic salts, sodium chloride, for example, doesn't get concentrated as much. And that's an important observation, because when cells climb to these high levels of intracellular salts, this can be very damaging to the cell. So you can see here in the presence of glycerol that increasing concentrations, as you lower the temperature and as ice forms and we get an increased and we have water transport out of the cell, and we've increased that intracellular concentration, the more CPA present, the less concentration effect that you have. So this means we don't hit these sort of toxic levels of sodium chloride concentration. So that's less chemical stress. So this is where even with a single CPA like Dmso, you get improved survival. If we just look at the green curve here, we get improved survival on the fast cooling rate side, and you get improved survival on the slow cooling rate side, and you can just drive the survival up across the board. That means you have more flexibility in the cooling rate you can use, and just more survival, more net survival, better outcomes. So quick recap of some of the ones that are in widespread use. We have dimethyl sulfoxide we were just talking about. There's also ethylene glycol, propylene glycol. I put the structures of these over here. You can see these are polar. They have they're small. They penetrate through the cell membrane. They are good hydrogen bond formers. They have a lot of similarities. There are others like glycerol, methanol, ethanol that are also used, but not as frequently as some of these other compounds. Okay, so on to non-penetrating CPAs, or non-permeating. I'll describe these in the extreme, but we generally don't use these alone. We use them in combination with permeating CPAs. But if we were to use them alone, as I said earlier, if you put cells in a 1 molar non-penetrating CPA solution, it's going to shrink. Water is going to come out of the cell to equilibrate this chemical potential. And we have this critical lower volume limit that we need to be careful about. If you add a penetrating CPA into the mix here, so let's say hypothetically we have 0.2 molar DMSO, we've preloaded into the cell, and then we expose it to the same amount of non-penetrating CPA, the amount of water that must leave the cell to equilibrate this chemical potential gradient, or this difference in osmotic potential or osmolality, is less. So we don't run the risk of exceeding this critical limit. We can stay within these osmotic limits. So these are just some extreme cases to show how they function, how they impact the osmolality of the extracellular and intracellular space, and therefore that driving force. Okay, so some of the non-permeating CPAs that we use, this is quite vast, and we don't have time to go through them in great detail, but I will say that we have 2 major categories. Those are the sugars, like sucrose, trehalose, raffinose, etc. And then we have the high molecular weight polymers, and this is by no means an exhaustive list, because when you start getting into proteins, the people are investigating a lot of different polymers in an investigational sense to see if we might improve outcomes. But what they have in common is they're all high molecular weight over here, meaning they don't increase the osmolality of the solution too much, but they're very good at increasing the glass transition temperature of the mixture and the viscosity of the mixture. So they have a physical suppression or a physical chemical suppression of the propensity for ice formation. These all form nice glasses when you go to really low temperatures. The sugars are smaller relative to high molecular weight polymers, and will assert some osmotic influence, but these are also known to have stabilizing effects on biomolecules. So there have been studies that have demonstrated how trehalose, for example, can stabilize proteins in solution and can stabilize membranes and protect against freezing and dehydration stresses. So this is what it looks like, I mean, when you put them together, and this is typically how they're used. In the absence of a non-penetrating component, so if you follow the dashed line, if we just were to expose cells to Dmso, for example, at a certain concentration, and Dr. Devireddy went through this, the cell first shrinks because of the gradient. Water is rushing out of the cell, and then as CPA comes back into the cell and pulls water back in, it swells back up. If you have an additional non-penetrating CPA, the extent to which it dehydrates is less, and so this can sometimes help you avoid osmotic stress. And sometimes you load things in multiple steps, sometimes you load it in one step, but these are your typical shrink swell curves of prevalent preservation formulations. Okay, so those are the types, and then I want to switch gears to our second learning objective, which is dealing with the toxicity issues related to Dmso. So Dmso has successfully enabled us to preserve hundreds, thousands of different cell lines. So it gives superior cryoprotection. But when this became an issue was when we started to cryopreserve cell therapeutics, and we started to put these things into the body, these formulations into the body. And there have been lots of reports of potential complications of doing so. Is Dmso causative? I don't know that the evidence is necessarily definitive, but there have been enough complications and enough concerns that there's a desire to move away from Dmso in preservation formulations. And of course, there's the issue of when we directly inject Dmso loaded cells into the body and they return to an isentomic environment, and the Dmso starts to come out, but water also starts to flow in at the same time and at a faster rate. There's a risk of swelling cells and causing damage of these therapeutic cells, and therefore reducing the efficacy. So that's something we certainly want to avoid. So one of the strategies then for bringing down this toxicity effect is just to wash DMSO out prior to infusion. And we just went through how this works essentially, but it's essentially adding a non-penetrating CPA, and the washout formula can help prevent that excessive swelling that happens. And so this is a technique we're using, is just to wash out prior to use. Without washing out in the absence of a non-penetrating cryoprotectant can lead to what I call a kaboom moment, where the cell swells beyond this upper critical limit, and we have damage to the cells. And this can happen in the body. If we wash out in the presence of a penetrating CPA, we can prevent that excessive swelling. Okay, so another strategy is just to reduce the amount of DMSO in the preservation media to begin with. And there are many reports of doing that. And I mentioned these various high molecular weight compounds that can serve to reduce the amount of ice that's formed, increase the glass transition temperature of the mixture, and generally impact both types of freezing injury. And I won't go through these in detail. I just wanted you to appreciate that there's been a lot of work done on a variety of different stem cells from different species. And we haven't removed DMSO completely. We can generally, with addition of these high molecular weight compounds, get the DMSO concentration cut to in half, generally, with a very good outcome. Okay, so finally, remove. We could remove completely by replacing DMSO. So instead of washing it out, we can just replace DMSO with something else in the preservation media and not use it at all. And I'll just mention one that's received a lot of interest is trehalose. So trehalose, as Dr. Devireddy said, is mentioned, is prevalent in these anhydrobiotes that are able to resist excessive stresses, excessive dehydration, et cetera. So it doesn't, it's manufactured by species that are anhydrobiotic, but mammalian cells do not have that adaptation. So if we want to use trehalose as an intracellular cryoprotectant, we have to be able to stuff it into the cell. So there have been lots of work using a lot of different techniques to try and achieve this. All of them are generally time consuming and require some high technology approaches. So they don't easily lend themselves to large scale or clinical, large scale manufacturing or clinical environments necessarily, but they're generally effective. If you can get trehalose inside the cell with any of these methods, they generally do provide protection against cryopreservation stresses. Okay, so I know I'm getting up on time here, so I'm going to go through this very quickly. One point I wanted to make is the importance, if you're trying to understand, if you're trying to test CPAs as alternatives and you want to get a sense of relative toxicity, let's say you're doing high throughput screening, et cetera, it's important to be aware of the biophysical environment that you're working in. So with typical chemical testing of compounds, toxicity testing of compounds, you generally expose cells to a concentration series to find your limits of exposure. So when you're dealing with things which are hopefully less toxic, those concentrations can climb to very high levels before you induce death. So it's important to understand the impact of solution osmolality on cell viability as part of that overall picture. So you want to know if you're actually dealing with chemical toxicity versus just lysing the cell because there are too many osmolytes in solution. So you can delineate that region by just determining these osmotic limits before you get started. You can do that with a basic sugar like sucrose and determine what these critical limits are. And then you know your zone. You know the zone that's chemical, that's inducing chemical toxicity versus the zone that's inducing osmotic injury. So some of the basic methods for toxicity testing, these are the ones you'll see a lot in the last few decades of toxicity testing of CPAs. We have our membrane integrity assays, good old hemocytometer, very simple. You can buy these at Walmart now, $100. You load up your you put a little bit of trypan blue in your cell sample, load it up into these chambers, and then you count. You count the cells that are blue have taken up the dye, you call dead, and those that haven't, you call alive. The problem is there's a lot of opportunity for false positives here. Cells can be transiently permeable as a result of just handling stress or cryopreservation stress or anything you've done to it that can take up the dye and not necessarily be a dead cell. An alternative is to do what's called live dead staining. And now there are kits that are not just live dead, but live necrotic or apoptotic. You can distinguish between the way in which it died. So there are double stains and triple stains. But basically there's a stain that will go into all live cells, and then there's a stain that will differentiate necrotic from apoptotic in terms of membrane characteristics. So they're great. You can buy them as kits. You get all these wonderful colors for, you get a lot of information out of them. You can use flow cytometry or fluorescence microscopy to do the counting, but it can be labor intensive. In either of these cases, you've got a gate with controls. You've got to calibrate the level of fluorescence so you know what to call a live cell and what to call a dead cell. And then, of course, there are the metabolic assays. These are based on growth. So you can grow out your cell cultures and add this dye called resizurin, or otherwise marketed as Alamar Blue. And it basically, a living cell will reduce this dye from this blue form to this pink form, resizurin to resirufin, and you can read it in a plate and you get very clear signals. You can read absorbance, you can read fluorescence, and you can do this in very small volumes. So this is good for kind of high throughput, but there's also the possibility for some false positives as well. This is something I've seen in my own work, is that prior to dying, cells can exhibit what's called a hormetic response, where they increase their metabolic activity right before death. And so you can sometimes mistakenly can think that's growth when it actually isn't. So you'll see a lot of these in optimization papers. I would say that whatever you're testing, you need a couple. Generally, you want one of these kinds of assays, one of these kinds of assays, and you need a functional assay. So whatever your cell is supposed to do, you need some assessment of whether the cell is still able to do that. So if it's a reproductive cell, can it still reproduce? If it's a neuronal cell, can it still transmit an electrical pulse, or what have you? If it's a stem cell, is it still STEMI, etc. So now I want to do a quick leap ahead to say that's the way we've done it. What are the opportunities in the future? I think, using these, we can learn a lot from the pharmaceutical world, where they do a lot of trying to find leads for drug targets, et cetera, and doing high throughput screening. And the Alamar Blue assay actually lends itself well to that, where we can look at viability as a function of log concentration and determine what's called this effective concentration that causes 50% death. You can couple data like this with structural information about the molecules you're testing in a QSAR format, where you quantify, maybe the presence of hydroxy groups leads to increased toxicity or decreased toxicity, et cetera. And we can learn more systematically, as opposed to just using trial and error approaches. We can also use new tools like molecular modeling that teaches us, that can tell us in more exquisite detail what the nature of the injury is. So here, for example, this modeling paper, they looked at different CPAs and looked at membrane thinning and pore formation, and how that might be asserting an effect on the membrane. So this can give you a phenomenological basis for why your LP might change in the presence of certain chemicals. And you can also go the other way around, where if you know the desirable features of your CPA, you can use molecular simulation, et cetera, to maybe drive up your TG so you can get into glass transition temperature range at a higher temperature. And you can look at additives that can maybe allow you to engineer a particular outcome. And so I just wanted to share the key resources that I referred to a lot in this presentation. I wanted you to have that available. And with that, I know I'm a little bit over time, and so I'll stop and take any questions.
Thank you, Dr. Elliott. It was also a very, very nice presentation. We have been learning a lot here today. So again, just to remind you, if you have any questions, please don't hesitate to use the Q&R window. We'll discuss all the questions at the end of this last presentation that we have now. And to finish this session, I would like to invite Dr. Charlie Hunt. It's a real honor for me to invite him to talk today. Dr. Hunt's scientific background is within the fields of cryobiology, cell and tissue banking. In 1978, he joined an MRC cryobiology group at Cambridge University, where his research interest was the cryopreservation of cornea, pancreatic islet, and arteries. In 1992, he helped found the East Anglia Tissue Bank. This was the first therapeutic tissue bank within the UK National Blood Service. There he was responsible for the heart valve, stem cell, skin, and amniotic membrane banking services. In 2003, he became operations manager for the then newly founded UK Stem Cell Bank, providing human embryonic and other stem cells for research and clinical applications, where he remained until he retired in 2017. He's a past president of the British Association for Tissue Banking, past committee member for the Society of Low Temperature Biology, and current member of the editorial board for the Journal of Cryobiology. Dr. Hunt will talk about cryopreservation of human pluripotent stem cells.
So I'm going to talk about the cryopreservation of pluripotent stem cells. It's just over 23 years since the first human embryonic stem cells were derived from the inner cell mass of cleavage embryos, with the human iPS cells derived from reprogramming somatic cells first reported around about 8 years later. Over the years, much of the research effort has gone into understanding the conditions necessary to maintain the pluripotent state and selectively and successfully differentiate these pluripotent cells into cell lineages of clinical importance. But in this time, only limited research has been undertaken on the cryopreservation of these cells. A number of factors shaped these early studies. The cells themselves are grown as colonies. You can see them here in the schematic. And in the early days, they were grown on an underlying feeder area, usually mouse embryo fibroblasts. And maintenance of the cells in the pluripotent state was difficult and quite labor intensive. The cells were prone to spontaneous differentiation in culture, and enzymic dissociation into single cells for passaging usually caused apoptosis and cell death. So techniques for passaging were developed which didn't involve dissociation agents, with the colonies being divided into pieces and transferred from plate to plate. This was so-called cut and paste technique. Another contributing factor for the route taken early on was the experience within the derivation centres of animal embryo cryopreservation using vitrification. So for these early studies, here, the Rubinoff, Zoe and Richard studies, they used a fairly simple and standard vitrification solution containing dimethyl sulfoxide and ethylene glycol, and sucrose as an impermeant protectant in the VS2 solution. Using these lower, less toxic concentrations of cryoprotectants necessitates ultra-rapid cooling. to attain the vitrified state, a state that under these conditions is metastable and prone to devitrification if temperatures are not maintained below the glass transition temperature. So these colony pieces or clumps were transferred rapidly through the vitrification solutions, VS1 for around about a minute, VS2 for 25 seconds, loaded into these open pulled straws and cooled ultra rapidly by, in microliter quantities by transferring these, plunging them into liquid nitrogen. Now these three studies really set the tone for much of what was to come over the next 20 odd years. These studies compared this simple vitrification protocol with a standard slow cooling protocol used by many cell banks, 1 degree a minute in 10% Dmso and 90% fetal bovine serum. Now, due to a number of confounding effects, the presence of cells in clumps, tendency for the clumps to fragment during thawing, presence of feeders, and the need for reattachment, the use of membrane integrity or metabolic assays was virtually impossible. And this led to a reliance on downstream assessment, sometimes the days and even multiple passages, as you can see here, post-thaw. And the emphasis was largely on demonstrating the main functions of the cells, the maintenance of pluripotency, the trilineage potential through teratoma formation, and rather than investigating cryopreservation injury per se. Nevertheless, the study showed that no matter how bad the results were with vitrified samples, and you can see here in a lot of these studies, although they recovered large numbers of clumps, most of these clumps had significant levels of differentiation. They were even worse when you looked at the cooling under standard slow cooling conditions, to the extent that in these studies assessment was often not possible downstream due to the lack of any surviving cells. So from these studies emerged an early paradigm that human embryonic stem cells could only be preserved by vitrification. And this was unfortunate in many ways, since there are significant disadvantages, I believe, to pursuing this mode of cryopreservation with cells which are destined for scale-up and clinical translation. Now, over the last decade, much work has gone into addressing some of these issues with vitrification. Still using the same vitrification protocols, attempts have been made to scale up the process with bulk vitrification of clumps and colonies, using cell strainers. As you can see here, these are self-made ones, but commercial ones are available. And minimal vitrification solutions are being used then to maintain the ultra-rapid cooling rates required. Other groups have developed surface vitrification techniques, either of whole adherent colonies by replacing the media with the vitrification solutions and the vitrification solutions with liquid nitrogen into the plates to maintain the rapid cooling rates needed. Latterly, two groups have developed what has become known as the twist technology. This is again surface-based vitrification using these small, well, essentially two of these small plates fixed together. One with a compartment in which the cells are grown under media. The media is then replaced with vitrification solution. The whole device is inverted, and the adherent cells with just a cryoprotectant film are then cooled rapidly by placing liquid nitrogen into the top container. These studies using culture matrices, which allowed feeder-free conditions to be used, were able to utilize live dead, staining methods and metabolic assays, and showed, certainly in the surface-based vitrification here, fairly good post-thaw responses. Not only high metabolic and viability compared to freeze-thaw controls immediately post-thaw, but also high plating efficiency and low differentiation in the cultures. And then downstream, there was maintenance of the pluripotency using various marker technologies, and also of trilineage potential using embryoid bodies or teratoma assays. So whilst addressing some of the perceived disadvantages of vitrification, notably lengthy processing times, low throughput, and for the twist process, some of the elements of sterility related to contact with liquid nitrogen, many of the other disadvantages remain. Moreover, I think with these techniques, the potential loss resulting from a temperature transient causing devitrification is likely to be more significant with these techniques, where vitrification of several hundred colonies per dish or strainer is involved. And it's going to be more significant than it is with, say, a loss of a straw or straws containing possibly 5 to 10 cell clumps. Despite these advances in vitrification techniques aimed at scaling up the process, it's still a process, I think, more suitable for laboratory stocks and early passage insurance stocks of cells, where the stability of the cell line is less robust. Now, further comparative studies tended to confirm the poor survival of cell aggregates and adherent colonies when exposed to slow cooling. Attempts to encapsulate aggregates were largely unsuccessful, whereas significant improvements have been reported, particularly for whole colonies and adhering to the plates, for cells encapsulated under a matrigel coat, and also for aggregates adherent to microcarriers, which were then encapsulated in alginate. The first real improvements, I guess, to reporting for with slowly cooled aggregates came in a number of studies which examined the influence of cooling rate and seeding temperature on survival following slow cooling. These studies suggested a significant benefit of controlling undercooling. The optimum cooling rate in these studies was somewhere between 0.3 and around about 1.8 degrees a minute, with a seeding temperature range in these studies optimum at around about -7 to -10. The recovery increased in these studies from around about 50% to greater than 80%. There was low differentiation, and downstream pluripotency and teratomer assays showed that these cells were comparable to the original material. But again, these studies only proved provided metrics based on morphology some days following thawing. Nevertheless, compared to previous studies where undercooling was uncontrolled, it pointed to the importance of controlling this often overlooked parameter. A recent study by Lee comparing aggregates and single cells in suspension seems to have shed some further light on the subject. This study used a number of viability assays at time points immediately post-thaw and following, mapping viability over the first 4 days in culture using various apoptotic assays and viability assays. And they also used, I think for the first time, certainly with stem cells, Raman cryospectroscopy, which was to try and elucidate the position of the ice within the cell aggregates. Now, these are graphs taken from Lee's paper. The first set of graphs strongly suggested that aggregates were more sensitive, aggregates here and to single cells there, in their viability after cooling and the seeding temperature with the cooling down to these temperatures before seeding. The results suggested that the cooling rates close to what is considered around about the optimum 1 degree a minute were from these results, what they found was that the optimum that was suggested by the previous studies, around about -7 to -10, was probably too low. The optimum being with Dmso and from these cells for around about -4. And you can see here that at the slower cooling rates, the amount of intracellular ice as represented by the ratio of the area of ice to the area of cytoplasm, was much lower at the slow cooling rates than it was at the more rapid cooling rates. In aggregates, there was also a correlation between the amount of intracellular ice and the rate of post-thaw attachment. If you look at cooling rates seeded at -8, so all cooling rates seeded at -8, and at the 10 degrees per minute with seeding at -4, you can see that there was quite high levels of intracellular ice. And under these conditions, with the aggregates seeding at -8 and seeding at -4, but cooling at 10 degrees a minute, there was virtually no post-thaw attachment. Interestingly, if you look at the post-thaw recovery using the viability assays, under these conditions, the recovery was relatively high under the conditions where there was no attachment, which suggests that there's a decoupling of these two metrics, so recovery and reattachment. Now, the mechanisms behind ice propagation and the sensitivity of aggregates, but not single cells, to supercooling remains unclear. It's been suggested by others that sensitivity may relate to water diffusion through cell aggregates in response to freezing, while ice propagation may occur via gap junctions and surface catalyzed nucleation events at the aggregate periphery. Whatever the mechanism for these phenomena, seeding temperature should be considered, I think, a critical parameter in optimizing slow cooling protocols for pluripotent stem cells, as it probably should for most other cells. Right, the next slide shows some work that was undertaken at the UK Stem Cell Bank on an iPS cell line derived there, which was grown under feeder-free conditions and then slowly cooled in the absence of any kinds of apoptotic inhibitors at 1 degree a minute in a standard Dmso FBS cryoprotectant. The cells were followed for 48 hours and subjected to live dead staining at the time points shown. The graph shows a typical post-thaw response of HPSCs with high viability immediately post-thaw, but this doesn't translate into attachment or continued viability over the following 24 to 48 hours. Accompanying these studies, we also looked at early and late apoptotic markers up to 24 hours post-thaw, and these were all elevated during in this period. Now, this and a number of other studies have shown a significant rise in levels of early and late apoptotic markers, and this loss of viability over the subsequent 24 to 48 hour period. It's a relatively common occurrence and has been termed cryopreservation-induced delayed onset cell death. Now, most perturbations on the cell by cryopreservation, from cell volume excursions to ROS production, are known initiators of apoptosis, and cryopreservation has been shown to activate both the intrinsic and extrinsic apoptotic pathways. Now, the extent of this phenomenon is dependent not only on factors linked to cryopreservation, but also others linked to the cell type and the quality of the starting material. So failure to address suboptimal cryopreservation and the level of this latent injury, and only regenerating the cell culture from the few surviving cells, will not only result in lengthy downstream recovery as you try and build up the cell population again, but also may lead to selection of subpopulations, which, though more robust from a free-thaw perspective, may exhibit undesirable characteristics. So, with the advent of feeder-free culture and the introduction of milder dissociation agents, we've seen that the early cut and paste methods have been largely superseded and replaced by passaging as single cell suspensions. Instrumental in this has been the use of the Rho kinase inhibitor to minimize dissociation-induced apoptosis. This inhibitor, the main one being used is Y27632, is effective in reducing cryopreservation-induced apoptosis as well. And a number of studies have looked at this. The caspase inhibitors themselves, although reducing in this study caspase 8-9 levels, did very little in terms of controlling or reducing the effects of apoptosis. Other studies using antioxidant, in this case the antioxidant catalase, although this is not an apoptotic inhibitor, it is effective in lowering reactive oxygen species, which themselves can trigger apoptosis. And the use of antioxidants, I think, can bear further study. Of the apoptotic inhibitors that have been investigated, so these are some of the ones here, BAX and P53, only really the ROC inhibitor has been shown to control this cryopreservation-induced cell death. And it's been most effective in all these studies when it was added to both the cryoprotectant and the post-thaw medium. Now, although ROC has been used quite consistently now over the last few years, and the studies are now showing that it doesn't increase mutation rates in pluripotent stem cells, there are nevertheless some reports of negative effects on extended use of ROC in culture. This seems to be a particular concern in iPS cells, and probably argues for a more judicial use of this inhibitor and limitations on exposure times during the cryopreservation cycle. Now, the ROC inhibitor Y27632 is a very expensive additive, but there have been, and is not actually licensed for clinical use, it's licensed for clinical research only. But there is a clinically approved cheaper alternative, Astudil, which has been shown to be as effective as Y27632 for cryopreservation and dissociation-induced or controlling dissociation-induced apoptosis in stem cells. So finally, I'd like to concentrate a little bit on, briefly I guess, on alternatives to Dmso. Now, we've already heard about CPA-induced toxicity and osmotic damage that's been demonstrated for a wide variety of stem cells, and this also is shown for HPSCs. And this kind of damage applies, of course, to all CPAs, not just Dmso, and can be mitigated by reducing CPA concentration, exposure times, and temperatures, as Gloria showed. And as she said, there's an increasing concern over the use of Dmso in novel cellular therapies from a safety perspective. The clinical side effects that she mentioned have been noted particularly for hematopoietic stem cell transplantation, ranging from mild to serious, and as she said, associated generally with direct infusion of unwashed cells. Now, in these situations, they are often mitigated by lowering Dmso concentration or washing before infusion. Since pluripotent stem cells are not likely to be infused directly into patients, adverse reactions are much less a cause of concern here. However, perhaps more worrying is the part Dmso may play in genetic and epigenetic changes to the cell. Through its actions, though its actions in this area are largely based on studies which see the cells exposed to Dmso in culture media at 37 degrees, often for hours or days, A recent study by Verhagen has indicated that effects on the epigenetics may occur within as little as 2 hours with concentrations which are substantially less than those that are used in cryopreservation. Although again these studies were at 37 degrees and with the DMSO added to the culture media, it does raise the possibility that we should be at least looking or moving away from DMSO for reasons other than adverse events. DMSO is also a potent inducer of differentiation, which is a concern for its use with HPSCs, and to a certain extent somewhat ironic, as the CPA now will often include ROCK inhibitors which inhibit differentiation. The table alongside here just shows a number of alternative cryoprotectants with either alternatives of DMSO-free alternatives or one where the DMSO concentrations are reduced. For the user, the problem with this is that the formulations are often commercially sensitive, and the CPAs, whether they're singular or plural, will not be disclosed. From a therapeutic perspective, this is can be a problem, but it is mitigated through the presence of a drug master file system where files on the CPAs are lodged with the FDA. Now, use of a CPA with a drug master file, of course, doesn't itself confer regulatory approval, but it can be cross-referenced in any submissions of a cellular therapy to the regulators. One development of note in the search for alternative CPAs is this recent paper, again by Lee, with a novel DMSO-free cocktail, which excludes the need for ROCK. It's a recent, as I say, a recent report, and it looks as if this may provide a alternative to the DMSO cryoprotectants cocktails that are currently used. Now, there are also alternative non-frozen storage options that have been reported for pluripotent stem cells. 7-day storage at ambient temperatures have been achieved using cell spheres. The cells are grown as cell spheres in E8 medium. This is a minimal stem cell medium, and good recovery of PSCs was seen 7 days post storage. Also, again, cells encapsulated in alginate in hypothermosol, which is a hypothermic, commercially available hypothermic storage solution. And one interesting report, but again, which has only just come out, is the use of what the authors term deep supercooling without any cryoprotectant, and they avoid surface catalyzed nucleation here through the application of a immiscible surface oil onto the top of the storage solution, and they've shown good post-storage function after 7 days storage at -13. So, just in summary, then. Early attempts at cryopreservation were shaped by the colonial nature of the cells and the problems of dissociation-induced apoptosis. This led to vitrification of aggregates or adherent colonies and becoming the method of choice, as the early attempts at slow cooling resulted in extremely poor, if any, survival. The problems with vitrification have been, attempts have been made to tackle these with methods for bulk vitrification, but these are still ill-suited to scale up and from a microbiological point of view have significant issues. Cryopreservation induced delayed onset cell death is now, I think, quite confirmed as an important player in the poor recovery of slowly cooled HPSCs without the introduction of any kind of apoptotic inhibitor. But the single cell suspensions that are now that we are now able to produce using the ROC inhibitors and the use of ROC inhibitors in the cryopreservation solution has now improved the post-thaw recovery of these cells. One important, I think, aspect of this, as I said, is that seeding temperature is a parameter that needs to be looked at more closely in these cells as with others. And future optimization of these cells should consider, in terms of the assays used, the tiered approach that's been advocated by John Baust and others. I haven't had time to go into this, but the papers by Baust provide an excellent overview of the advocation of this approach. I think also further research is needed on genetic and epigenetic effects of DMSO exposure pre and post cryopreservation, and I think we need to consider more closely DMSO-free alternatives where possible, not only for HPSCs but for all cell therapy products. So I think that's me done for now.
Thank you, Dr. Hunt. It was a great talk again. We have a limited amount of time now for the questions. So I suggest that, like Professor Devireddy and Dr. Elliott did already, Dr. Hunt answer the questions in the Q&A window, so we can move forward for the next session.